Hey everyone, and welcome back to AP Biology. In our last lecture, we talked about speciation, and we brought up Darwin's Origin of Species. We talked about reproductive barriers, allopatric speciation, and sympatric speciation. In this lecture, we're going to go over the next standard for AP Bio. We're going to talk about where the origin of life came from. Okay, something called biogenesis. Okay, if you know from the Bible, Genesis is the first chapter. It is the beginning. So what is the beginning of life? Now, as far as we know with the fossil record, life started 3.5 billion years ago. The Earth began 4.6 billion years ago, and the universe itself is over 12 billion years old. So we're looking at this as a really far out thing, a lot of hypotheses, very few theories, and no laws. But in life, we have to have some sort of groundwork before we move on. There were two scientists who were really curious about this, named Miller-Urey. Okay, but first let's talk about Stanley Miller, because Stanley Miller was the one who came out with this machine. Basically, he wanted to mimic what early life would look like. In early life, we had a reducing atmosphere, which means that there was no oxygen. We have proof of this from our rocks. There's certain iron ore rocks that should have rusted maybe 4 billion years ago, and it didn't. 3.5 billion years ago, and it didn't. How come we don't see rust? When oxygen and iron come into contact with each other, they make iron oxide. So knowing that, we're going to add some gases into his chamber. Okay, this is where the reaction chamber was, and this is going to stimulate lightning, because lightning gives away free electrons, and when we have a reducing atmosphere, you're adding electrons to the molecules. And so let's take a look. So here's this experiment right here. We're going to put all these wonderful gases into this chamber. But before that, we're going to use water. Because one thing we know is that the Earth had plenty of water before life ever began. And we also know that cells need to be submerged in water to actually survive, right? So what we're going to do is we're going to heat up the water, and it's going to come through this little tube. As it comes, the gas comes through this little tube, it's going to have to enter into the reaction chamber that we just saw is going to simulate lightning and electricity. We have water, we have methane, we have ammonia, hydrogen, and carbon monoxide. Basically, all the atoms we need. The only thing we're missing here is phosphorus and sulfur. But we do have C, H, N, and O, which is beautiful because you make most organic compounds. Now we're going to use this thing called a condenser. That's going to send in cold water and cool the gases down. That's going to cause condensation when when a gas turns into a liquid. Now we have a liquid coming out, and then what we're going to do is we're going to trap that liquid and see what happens. This sampling probe proved a few things. Every time we did this, something different happened, but we did get amino acids. In some cases, we got sugars, and in other cases, we even got small chains of fatty acids, which means that life can start as long as this is in a reducing atmosphere. Just means that organic molecules can form based on these very simple things, maybe from the energy from a meteorite or the energy from some lightning. All right, so Stanley Miller wins the Nobel Prize for this. That's a big deal because it tells us where biogenesis happens. Okay, so what I just said before, this has to do with a rock layer called stromatolite. And what we notice is that there's a rust layer, but then before that, there is no rust. This is how we know there was no oxygen on the Earth at 3.5 billion years ago. We start to see the oxygen about 3.0 billion years ago. All right, and this is where we see it. Right now, the iron looks untouched. Okay, it has not rusted. Beautiful. Now, another thing we've made in the lab is artificial membranes. We've made liposomes and micelles. The only difference between the two of these is this is a phospholipid bilayer. So just by putting phospholipids together, you can make these tiny little vesicles, okay? Tiny little fake cells. Or you get a micelle. When you do that, that's just a monolayer of lipids, but nevertheless, as long as the tails are hitting inside and they're hydrophobic, they're scared of water, they'll be fine like this. This is what we really see, okay, when we look at a lipid bilayer. And so this looks really nice. By doing this, we can now create something called a liposome. We're going to create eventually something called a protocell, or a fake cell, where we have like reactions going on, but it doesn't have the DNA, it doesn't have the ribosomes, it cannot reproduce and function on its own. Nevertheless, it can still do reactions that are different from the outside environment. And so liposomes was the first thing we did, right? So with these protocells, little droplets, imagine this, taking a liposome, throwing a few enzymes in there. Let's put something like maltase in there. Then what we'll do is maltase breaks down maltose into glucose and glucose. So let's say we brought maltase in, the enzyme works on it, breaks down glucose and lets them leave. Wow, this cell is like an enzyme on wheels. It's beautiful. If you put more enzymes in there, you'll have like a nice little hard vesicle, similar to what a lysosome would be. Still, we don't have cells yet. But before we get there, you're going to need DNA. You're going to need genetic information. We know that the Earth was an RNA world long before it was ever DNA. And the fact that we know this is because of the number of RNAs that we have. We have messenger RNA, transfer RNA, ribosomal RNA. We have heteronuclear RNA, small interfering RNA, microRNAs. Okay, we have SN, or small nuclear RNAs, but we also have SNO, small nucleolar RNAs. So I already named like maybe 7 or 8 RNAs. The fact that they're still in cells today and there's only one type of DNA definitely gives evidence, strong evidence, that this was the first thing that ever arose. RNA world, okay? Very simple. And RNA could be single-stranded, but in some cases, like in viruses, we found them to be double-stranded. Knowing that, we've discovered something called ribozymes. Basically what a ribozyme is a catalytic RNA. It's an RNA that can do something like a lot like what an enzyme can do. Simply by putting RNA and protein together, you have this awesome machinery that can catalyze reactions. One great example is the spliceosome that cuts out introns out of a newly made messenger RNA in eukaryotic cells. The spliceosome is really just small nuclear RNA mixed with some proteins. Without that small nuclear RNA, there is no catalytic ability. Now, we also know currently we have enzymes, which are completely all protein and have no RNA in it. But before enzymes, there were ribozymes. It looks a lot like I'm saying the word ribosome. A ribosome is similar to that. It does catalyze the reaction of protein synthesis, so we could think of a ribosome as one of the first ribozymes. Knowing this, we also have a great way of dating fossils. So this is more of evidence for evolution, because this definitely comes in there, right? We have a lot of isotopes that are unstable because their nucleus is so heavy. When you have a very big neutron to proton ratio, like for example, if it's greater than 1 to 1.5 proton to neutron, you get this unstable nucleus occurring. Uranium is definitely one of them. Carbon-14 is definitely another one of them, uranium-235. And so they have this thing called a half-life. Now, this is not talking about any specific isotope, but what it is showing you is that every half-life that occurs, there's only half of this isotope left. And so another half-life occurs, and now there's only a quarter left, 1/8. Nevertheless, this is still a limit that is going to approach zero but never quite make it there. So no matter how old it is, we could still figure out the age simply by using this carbon dating. Now, the other thing, we eat a lot of carbon-14, whether you know it or not. About 1.5% of all atoms of carbon are carbon-14. Now, when we're alive, we keep taking in more carbon-14, but when we die, we don't take any more in. So as we die, our carbon-14 begins to decay every 5,730 years. When it does, a half-life has just came, and then half of those carbon-14s become nitrogen-14. So we could tell the age of a fossil simply by measuring how much carbon-14 is in it and then comparing it with something that's alive today. And so we have a really good idea of this absolute age of fossils. Another thing that we notice is that from this fossil evidence, we're able to create a geological record where there's 4 major eras because they're mapped out by these mass extinctions. So when we're looking at the history of the Earth, you should know these 4 things. Eon is the biggest, and they're broken down into eras. The 4 eras are broken down into many periods. Those periods are further broken down into epochs when we get more closer to the current date. And so this is a great picture of this. Okay, now really I'm only showing you half of the story. Okay, where we have the most recent Cenozoic era, this is today, and humans show up around right here. 1.8 million years ago, Homo sapiens shows up. The first origins of Homo show up about 5.3 million years ago. And if we get even further back to like 6 million years ago, we see Australopithecus and Homo living together. All right, we also have the Mesozoic era, right? So we have 4 eras. Cenozoic is the most recent. Mesozoic is the one previously after us. This was the age of reptiles. This is where dinosaurs ruled the earth. And there are 3 periods, starting earliest at the Triassic, then going to the Jurassic, and then eventually going to the Cretaceous period. Okay, and flowering plants begin to take over. Here's the other half of the story where we have something I like to call the Precambrian era, because right here, the Cambrian period is a big thing. Sudden increase in the diversity of many animal phyla. An underwater world begins to emerge. Before that, we start to see a lot of unicellular organisms during the Precambrian era. Okay, and so just think about this Cambrian explosion about 500 million years ago. This is an artist's rendition of what the Earth looked like, actually underwater, where we see trilobites. We saw these long worms that don't even exist today. Some crazy organisms like we see over here. Everything in this picture, probably except for the sponge, is extinct. Okay, the only one alive today, I would go with the sponge and some of these species of algae that are really small. But then go to the next big event in the Paleozoic era would be 350 million years ago, where vascular plants first show up. Okay, and this is also another big deal that we're seeing, because now plants came from water environment to a land environment. Life is beginning to take over. We're starting to see life on land. All of a sudden, the animals are getting more bold. We start to see crustaceans show up on land. The evolution of insects show up. Okay, so now we're looking at these 4 major eras, and I just went over the Paleozoic era. I went over these 2 eras, and we said that this was the unicellular world. So just a recap, unicellular world. Over here, I'm going to say this is the origin of plants and the origin of the underwater world of invertebrates. Okay, and at that same time, invertebrates do make it to land in the form of insects. During this era, we have the age of reptiles, and dinosaurs rule the world. And this one I like to call the age of mammals, because now we start to see a huge diversity and jump in the mammal department. Okay, now another thing you have to know who Lynn Margulies was, because she came up with the endosymbiont theory. When we get to the chapter and we go over cells, I'm going to talk about how mitochondria and chloroplasts were once free-living bacteria. But I think this is a good time to bring it up, because we notice that mitochondria has 2 membranes. Strange, because what organelle should have 2 membranes? What's to give you that one doesn't? This must be evidence that you were once engulfed in the process of phagocytosis, and your second membrane, so that's what we're seeing here, the mitochondria was swallowed and received a second membrane. And then the same thing happened with the chloroplast. So in this example, I'll call that an animal cell. And in this example, the origin of the plant cell. Okay. Another thing is tectonic plates played a big role. You have to remember there was continental drift at one point. We had Pangaea. Then eventually we had like Eurasia and a bunch of, I forget the other names of these continents, but this is what we're seeing. These are the major plates. So whatever is outlined in black, as you can tell, a lot of the tectonic plates are underwater. We have convergent plates that come together and make mountains, and we have a lot of underwater mountains. So, for example, between the Atlantic Ocean, there's this Mid-Atlantic mountain range that's the biggest mountain range in the world. It's bigger than Mount Everest, okay? It's really, really tall. In addition, when they spread apart, the ocean floor spreads and we create deep sea trenches. All of this plays a big role in moving these continents around. All right, so this is what we see here. So this is what I was talking about. You had Pangaea, then you had Gondwana and Laurasia, and then eventually you get Eurasia, North America, South America, Antarctica, Australia, and India. Eventually, India smashes into Eurasia, and you get the Himalayan mountains as we're seeing here. So India is going to move up and bam, create that little tail on Asia, Southeast Asia. Okay, another picture of this as well. All right, mass extinctions always play a big role. I don't know if you guys know this, but 99% of all organisms are extinct. Okay, so really today we only have 1% of the 100% of organisms that ever lived on this planet. Less than 1% are still alive today. 99% are all extinct. And this is a big field because we're looking for these new fossils. We're looking for new evidence of evolution. And so here's some just major periods where we see that in the late Triassic. This right here had to be the dinosaur extinction. One of the biggest ones that we know, but there was definitely a big one in the late Ordovician period, the late Devonian, and the Permian Triassic period. Not to mention the Cretaceous Tertiary. I actually think this one, I'm sorry, not late Triassic, this would be the dinosaur extinction. There was definitely an extinction that happened a little bit before that. Now, what were these extinctions? Well, the impact hypothesis was this. Things that happened today can still happen. They still happened a long time ago. Now, of course, the way the dinosaurs went extinct, we know that a huge meteor hit the Earth. When I say huge, I mean really, really big. This thing created all this dust and uplift. Okay, that came. Now, if you have this thing hit the water, you'll get this huge tidal wave. That could have killed them as well. But there's evidence that it hit land, created this huge uplift of smoke that lasted maybe 60, 70 years, which is definitely enough time to kill out the whole species of dinosaurs. Mammals then, in the size of like this big, like a shrew, came over and ate the dinosaurs, lived off the dead flesh, and then they're still alive, enough to make evolution happen for the other million species. Okay, and so just taking a look at over here from our adaptive radiation, right? This is what we said when organisms radiate. You start with one species and they radiate outwards. We start to see this reptilian ancestor give rise to an ancestral mammal. That ancestral mammal gives rise to monotremes, which only 5 species are around today, marsupials with 324 species, and then about over 5,000 different eutherian placental mammals. These are just mammals that have their own placenta. They don't have a placenta because they have a pouch. These guys don't have a placenta because they lay eggs. Different kinds of mammals, yet we're seeing this diversity of life. Okay, one of the coolest things that we saw as well was paedomorphosis in this whole evolutionary scheme. We've seen that organisms like this one, it's called the axolotl. The axolotl will actually keep its juvenile features well when it's an adult. It keeps its gills. Hey, you're an amphibian. What are you doing? You should have become with lungs by now. But he's like, no, I like being like this. I live on the water. Cute? Yes. Amphibian? We're not sure. Amphibious? Probably more. Okay, and so when we think of the evolution of the human skull, this also comes into play. So we start to see the mammalian skull, right? From the human fetus to the chimpanzee fetus, there's a lot of similarities. Isn't it amazing how they actually develop into totally different things? Okay, but this is just an example of how divergent evolution takes place. Okay, finally we get to the last part of this, is the heterotroph hypothesis. It is believed that the very first cells that were on the planet were heterotrophic, which means that they ate each other. Okay, they weren't able, and if they were autotrophic, we would see evidence of oxygen, which we don't see. So this tells us that cells used to just actually engulf each other and eat each other. And then this makes the most sense. Okay, in this reducing atmosphere, that's what we had. And that makes sense, and it lines up with Lynn Margulies' endosymbiotic theory, where the mitochondria was swallowed up. Hey, don't digest it right away. I think this thing gives me more energy. Let's keep it. And that's how the mitochondria got its thing. Oh, don't digest this chloroplast right away. Why? Because if I leave it on, it feeds me for free. Let's keep it. So cells that did that and didn't digest the mitochondria or the chloroplast live on today because they had an advantage. Heterotroph hypothesis. Okay, guys, this concludes our lecture on the origin of the Earth and the origin of life. In our next lecture, we're going to go a little closer into taxonomy and start to look at the 5 kingdoms. All right, guys, so keep on reading that textbook. And until next time, I'll see you later. Be safe.